Okay, guys? Like you're used to, right? Okay. Okay. All right, ready to go. So welcome to the Biodiversity Boutique. My name is Professor Laurie Foster, and I'm very pleased to have you here today. And our focus that we're going to spend our time on is to work on DNA. Are you familiar with what DNA is? Mm-hmm. Do you know what DNA stands for? Go ahead.
Deoxyribonucleic acid.
Wow, that is a mouthful. Very good. What do you guys know about DNA already? Tell me what you know. Go ahead, just randomly. What do you know?
Raise your hand. Kind of like controls what people look like.
All right. Okay, all right. It governs how they look and some of the things they can do.
It exists in the nucleus of a cell.
Ooh, it does indeed. Uh-huh.
It's a specific sequence of nucleotides.
All right, good.
Sheesh, you guys are gonna know more than me.
Uh-huh, very good. What else? Do you know what those nucleotides are? What their basis? Oh, good. Let's hear about it.
I wrote it in my simple way. Any group of organic compounds composed of one or several nitrogen bases linked to a sugar and a phosphate group.
All right.
Hit some points that we're going to talk about later.
Fabulous, fabulous. Well, very good. So I heard you say sugars and phosphates in backbone, and that is right on target. I heard you say that it helps cells do things, so we'll put that one up there, that it helps organisms do stuff. Have you heard of anything called like DNA fingerprinting? Does that sound familiar to you? What do you know about that? Anything? Just heard the name? Mm-hmm. Are you familiar with the word?
Is that where you identify someone by finding out with their DNA?
Exactly, using their DNA for identification. Uh-huh. Was that what you were going to say as well?
9/11.
Mm-hmm,
You
exactly.
watch any of those crime shows and they figure out who does the crime thing by their fingerprints or what they touch? That's a huge factor.
So there's this thing, it's got a fancy word, it's called biotechnology. Heard that word before? So DNA is used for all kinds of things. These are just some pictures of some things that DNA takes hold of. So it can identify a person. We can take DNA and put it in other organisms and give them some genes, some things that they didn't have in their information packet. And eventually, we can look at identifying who done it, who was a criminal, who committed a crime, if their DNA was left at the crime scene. So we can do all kinds of stuff with DNA. Yeah. So it gives us this instructions for life, and it's a code that really is quite almost magical, but it's not magical, it's scientific, right? There's a difference, isn't there? All right, so what I want to do today, we have 3 things we're going to work on. I'm going to show you a quick little video, about 4 minutes long, that I think you'll enjoy. Then we are going to pull some DNA out of your cells.
Awesome.
Uh-huh, and I promise it won't hurt. You don't have to draw blood. Oh, that's too bad, huh?
I don't want to draw blood.
But, you know, blood is one place we could get cells from, but my goodness, where else are cells on your body?
Does earwax have DNA?
You know, it probably has some cells from your ear canal, so you could probably find some if you needed to look through earwax. What else?
Your hair.
You could find it in your hair, because what is your hair actually?
Dead skin.
Dead skin cells, exactly. Well, another one you had?
Saliva.
From saliva, that's what we're going to use. What's in saliva? Is it the saliva that we're looking for the DNA in, or not?
It's the cells that make up the saliva.
You've got it, you've got it. What else do you have?
Your finger.
Oh yeah, well, you know, your fingerprint is uniquely you because of your DNA. But, yep, but we probably wouldn't be able to get cells directly from a print, though, because we're taking a print like from some dye or some ink, and we're putting that down to show us the folds in our finger itself. But it sounds similar, doesn't it? Like it's a fingerprint. Let's go back to the saliva idea. So in a few minutes, I'm going to have you take some of your, not just spit. what we're going to do is we're going to just take a little bit of water and we're going to have you hold it in your mouth for a whole minute. And that may seem like not long, but a minute in your mouth is a long time. And we're going to just swish it all over, back and forth.
We can't look at each other because we might laugh.
And we better not laugh. That's right. We better not look at each other because we'll be laughing. And then what's going to happen, you tell me. What do you think that swishing is going to do?
It's good. It'll, like, the cells will rub off into the water, and then we'll be able to get the cells.
That's right, we are. We're going to get the cells from the water. Is that your thought too? Well, all right, good. So then we're going to give them some treatments, and then in about 15, 20 minutes, we can actually, with a few simple chemicals that you have at home in your kitchen, baking soda, salt, dishwashing detergent, they can come together and actually help pull the DNA right out of your cells.
So no super complicated.
No super complicated procedure. Isn't that cool? So you could, in a way, you could probably do this at home sometime. Okay, so we're going to do that. So we're going to watch a movie, we're going to extract your DNA, and then Crystal, tell them what you're going to do.
Well, we're going to talk about DNA and kind of hear a little bit more in detail about what it is and what it's made up of. And then we're going to make some super sweet bracelets. I have one on right now. And don't worry, I have ones for the boys if they don't like the beads. So we're going to be making this, and this actually is a code of my DNA spelled out from my name. So this is specifically me, and each one of these represents something from my DNA. So we are going to explain that to you guys a little bit later. Then after that, you guys gotta have your listening ears on, and we're gonna be playing a review game. So it's gonna be kind of like a Family Feud. Has anyone ever seen that show? When they go up against each other and they compete, we're gonna kind of split up, and there'll be a good prize for the people that win. All right? Okay. No, to split up.
All right, so
Let's get started with the movie.
all right, sounds good.
Your body is made up of 50 trillion cells. Cells come in many different varieties, with many different functions. But inside almost every cell is a nucleus, containing 99.9% of your genes, and mitochondria, containing a few more genes. All told, you have nearly 20,000 genes. Your genes are small parts of a long molecule called DNA, or deoxyribonucleic acid. If you lined up all of the DNA containing all of your genes, it would measure 6 feet long. But it's coiled so tightly that it fits in just one cell nucleus. DNA is a double-stranded molecule composed of sugar, phosphate, and 4 different bases: adenine, thymine, cytosine, and guanine. These bases spell out the language known as the genetic code. The number and order of these 4 bases determine, for example, whether you are a chimp, a cow, a banana, or a human. Most genes are recipes for making specific proteins. These recipes are passed down from parents to children, from generation to generation. When someone says, "You have your father's hair," what they mean is, you appear to have inherited a gene or genes from your father that makes a protein that instructs your hair follicle cells to produce hair that curls like your father's. But they usually opt for the shorter version. Genes tell a cell how to function and what traits to express. More specifically, gene regulators turn different genes on and off in different cells to control cell function. The long molecules of DNA containing your genes are organized into pieces called chromosomes. Different species have different numbers of chromosomes. Humans usually have 46 chromosomes, 2 sets of 23, or simply 23 pairs of chromosomes. Chimpanzees have 2 sets of 24, or 24 pairs of chromosomes. Rhesus monkeys have 21 pairs of chromosomes. Cows have 30 pairs of chromosomes. Chickens have 39 pairs. Fruit flies have 4 pairs. And bananas have 11 pairs. So, what percentage of the DNA in your chromosomes do you share with other species? You share 93% of your DNA with the rhesus monkey and 98.5% with our friend chimpanzee. How about with other humans? 99.5%. So, what makes us different from one another? Well, for one thing, SNPs.
And that's where we're going to stop this show. And these are really fun, so I can certainly send this information to how to find them. There's 4 of those shows in there. Did you like that? Yeah. It's kind of informational. All right, well, we are going to go ahead and get some DNA from your cells right now. And we need some time for it to process. And then Ms. Crystal's going to come back and do some more work with you on your DNA bracelets and learning more about DNA while we're waiting for it. All right? So I'm thinking, let's see, with the 8 of us, we can go to maybe 3, let's go to 3 tables. And we're going to, there are instructions on the table, and we've got some helpers here who are going to help us. All right? So
Let's get your saliva.
Here we go. So our first thing you have to do is kind of follow that recipe along, Courtney, Crystal, and Matt, if you could kind of help give them a sense of that. And we don't need goggles on for this thing.
Okay, so we're just going to follow the recipe.
We're going to follow the recipe, yep. So you'll want to follow the recipe along. I can't talk because I'm on a microphone. They're videotaping us.
So we have 50 milliliters here. We need 100 of those, so we need to fill it up twice, right?
Okay.
So we're starting here, right?
We've already added the 100 mils to the
Oh, you already
beaker,
have it.
so
Okay.
that part is
Okay,
done.
all right. So
Yeah.
we're on number 2.
So we've got the 100 mils already added, so that's done here.
So you're always,
or is it this
you're always
First part
constantly.
here.
Okay. Whoa.
You're constantly.
So this is your 100 mil beaker. That one's already done, part 1.
Perfect.
And then we'll just continue
So
on.
this is the buffer solution's already made?
Well, you're making the buffer solution.
Oh, okay.
It's ready to get started. Continue on.
Okay, gotcha.
Uh-huh.
We need 1 teaspoon of baking soda.
So we're gonna drink the water after.
Teaspoon
Not that water. You're going to drink some of that water.
salt and baking soda. So we're gonna need the 1 teaspoon of salt.
So 1 teaspoon of baking soda. And stir it so we get it a little bit more level. Take 1 more. Go ahead. Just tilt your jar a little bit. There you go. Okay, now make it a little level. Oh, there you go. Okay. I guess that'll work. Okay, and stir until completely dissolved. So stir it again. And now we're going to put in 1 teaspoon of detergent. So we got to find the detergent. Is that it?
That's it.
So 1 teaspoon of
You have
that.
to, there's a little dropper here. You have to unscrew the dropper, honey. There you go.
Is this the pineapple juice?
Yep.
Ooh,
Yep.
cool.
Yeah.
Fresh.
Fresh.
Yeah, use the dropper and measure it in here. Yeah. There you go. Perfect.
So how long have you been teaching at the college level? Or?
So, well, I've been teaching here 24 years. I taught high school and middle school for 6 years. So it's my 30th year.
They're still
I'm like 20
Are you?
years. Years off to have kids.
Yeah. Oh, it's still a joy. I still love doing it. So.
Do you want to switch?
Dynamic because you found they are all individuals.
Oh, they are indeed.
See, there's still tons.
Let's see. Oh, it's looking pretty good.
Okay, how we doing on our buffer solution? Looking like it's ready? Got pineapple juice ready to go.
Just one little drop, right?
Mm-hmm.
For the pineapple?
Yeah, 1 drop. Mm-hmm. And even if there were 2, it's fine. But one is fine. All right, we're waiting for our detergent.
Is our detergent man.
Okay.
Okay.
Let's see.
You need a paper towel.
Can wash them later, huh? All right, stir that detergent real good in there so it's, you got it, looking great.
It'd be fun to do a take-home project.
All right, 1 drop.
Oh, look at that
One
one
or-
drop. No, that's not much, really.
No, that's about all it needs is a little drop of pineapple juice. It's got some proteases in it to help strip the proteins off the DNA molecule.
This is enough for 4 people.
That will be enough for 4 people. And then once your buffer is made, you're going to go to part 2 then.
All right, so no, you're not swishing that in your mouth.
No, you're not putting that in your mouth. No, no. All right, so you got your buffer made up.
What is this?
And so now we're going to get our cups and get just a small amount of water.
It's just like you're using mouthwash.
All right.
Not a big switch.
Just like about that much. Okay? I'll just do this part for us. That good or is that too much?
You don't have to use it all.
I don't want to get too much, then they can't swish. That should be enough, honey.
Okay.
And now you need to let it swish in your mouth for 1 minute.
1 minute.
1 minute. Okay?
All right, I got your time, okay? Put it in.
That much?
Yeah. Swish away.
All right, into this.
All right.
So I'll let you know when it's about time.
So you gotta,
All right, Miss Courtney. Whoopsie. That's all right. That's just some water. No problem. There we go. You giving it a try too?
Absolutely.
Good. Miss Crystal, do you want to do it too? Grab a cup and we can have you give it a try. Dr. Matt, are you going to try it too?
I'm passing actually, because I'm an alien.
He doesn't have DNA. He uses arsenic.
I have the arsenic. Exactly right.
Swish, 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 real vigorously, back and forth.
I followed it.
Oh, you did? All right, swish away for 1 minute. It's a long time. A, B, C, D, E, F, G. They say that 1 minute takes to sing that song.
Do we need pretty much 20 mils?
Yes.
Okay.
Yes. And then approximately 20 mils of buffer into each test tube.
Yep.
This?
Yep.
And then did you spit in the test tube? All right. Oh, you need more test tubes.
Okay, this is mine at this end.
Alcohol down the side.
Not that kind of alcohol. This is isopropyl. Not, well, not in here we're not. And this is not alcohol that you would drink.
No, I mean from the
Oh,
water thing.
you know, it's probably not good in a laboratory to drink foods and beverages.
So we pour this in
All right,
here and
and then, yep. And then did you add your buffer? You got to pour your buffer solutions in as directed here. Okay, so take a small amount of distilled water in your mouth for 30 seconds. Pour 20 mils of,
Well, that's the 20 ml.
so 20 mils of buffer solution now into each of your test tubes.
Okay.
Now, okay.
How do you know?
All right. So you have to use your
that's what I
graduated cylinder. All right. So you're going to pour 20 mils of your buffer into each test tube now.
20?
Yep. And if they want to, they can probably, you guys want to pour yours?
Yeah.
All right. Miss Crystal will get hers, and then she can send it to you guys. All right.
And fill
I'm going
up.
to pour this in here, 15 mils for each person. We've got to hold it real slow.
Yep, and just pour it down the side.
Down the side, then we can put it in the rack and then watch it.
Exactly.
Now, this
All right.
So I'm going to pour 15 milliliters.
All right, so add your buffer. Okay.
Yeah, we can do that.
All right, put your 20 mils in.
measure carefully and slowly, okay?
Yep, real slow is the key. Oh, not that yet, honey. The isopropyl is going to be used later on. Right now we're using buffer.
But she gave me that.
Oh.
Sorry, it was my
Oh, yeah, no. So follow, yes, each of you take 20 mils of that buffer and pour it in your test tube. Okay, this comes later. We're going to use it, so just hang on. And you'll be ready for the next one. Yep, 20 mils.
You get down right with your eye, looking at it. It's good.
Got it? All right. All right, the next 20 mils for someone.
Should we stir this now?
Yep, stir it gently.
Gently. Very, yep, that's good.
About 15, I think.
All right. And when,
Keep track of your stirring rod.
okay.
Cup.
And there is a wax pencil if you want to label your test tube, as long as you guys remember. Okay, super.
I'm watching TV for a month.
Now stir it with your little stirring rod.
And then keep track of which one is your test tube once you get it in the rack.
This side, you want to put yours, or do you think you get?
No, I haven't put it
He's going to pour his 20 mils in now. Okay, you can get the stirring rod out of there. And now, after this, Crystal, we're going to take 15 milliliters of cold alcohol, rubbing alcohol, and you're going to pour it down the side real carefully and not stir it. Okay? So that's our next step.
This is yours. Okay, now, here's what it says. Let the mixture stand for 10 minutes. So 10 after, you will still see strands
Now
of
the
DNA
reason it's going to sit on
flowing
top
towards
is
the
because
alcohol.
it has a
So you
little
see
bit
the
different
layers right
solubility.
here?
So it's not
What's
going
gonna happen
to go
is
into the water
the
and mix
DNA
like the stuff we just poured in,
The
that buffer
DNA is
mixed
going
in with
to
the water.
migrate up.
The isopropyl alcohol that we're going
And
to
when it
add,
reaches the
it's going to sit
alcohol,
on
it's gonna
top. So you want to add it really slowly because
Very
it will mix a
good.
little bit.
And it's
Right.
cold.
It is
And
very
it's cold.
cold to keep it slower to move the molecules around.
So here's the 15 mark. Go ahead and add it to
All right, and we're just going to pour it on along the sides real slow. So see how it's floating? The alcohol is lighter weight than the buffer solution.
Perfect.
Very nice.
You want to do it yourself?
All right, 15 mils.
You might want to put this in here.
Yeah, in your rack.
We're
Yeah.
going to put yours right there, okay?
I'm gonna put mine.
Okay. Mine has this stir stick. Yours doesn't.
Okay, just kind of remember where they are. Actually, you want to pull that stir stick probably out even. Yeah. There we go. That looks pretty good. He thinks it's got to have one more little bit. And then the trick of the trade that works real good is if you tip this like that and just pour it slowly down the side. Okay? So yep, that's yep, and now lift this one up. Keep going. Keep going. Already starting out? You're perfect. You did it perfect. Just keep pouring and see how it's floating, you guys? Awesome. Pretty cool. And then we'll get yours ready to roll. 15. That one had buffer though, so we don't want to mess with that. Would contaminate it, wouldn't it? Looks perfect. All right.
Do you see anything?
Yeah.
All right.
Oh, now it's supposed to be worse than before.
So we're just going to let that set. Now very carefully put it back in there, and that's just alcohol. That's a rub, like, you know, if you get a sore, a cut, you can put rubbing alcohol to clean the wound.
Whoa.
All right, got about 15 or a little extra. Just a little titch too much. Let's pour the extra right in a cup here. That's fine.
It's the little right there.
Bless you. Bless you. And then you saw his technique, right? Okay.
That's you. That's the instructions for you. We can make more of you and put you in a spaceship. Yeah, yours is really kind of cool because.
And that's Emma's.
Yeah, look, see it's going up.
And that's Gabby's instructions.
All right. So we're all good over here? All right.
Laurie, we have Gabby in a test tube.
Oh,
What do we do now?
do we have, oh, we do?
Yeah.
Gabby.
Look at my instructions.
Look at that.
Gabby
It's
in a
already
test tube.
forming.
Mine have good
And that's pretty good.
Gabby in a test tube.
Yeah.
Mine is low. This is me in a test
Is
tube.
that
Look at this.
any
It's a-
fun?
we have
It's
a
working
ton down
already.
there.
Look at that. So we'll just let them settle, and the longer we wait, the better our product will be
Even
here.
going up here.
Very cool.
Yeah, it's nice.
All right. Come home with the
come home with
clone.
You can go clone yourself now. You can do that.
Remember how you guys had reactions with acid and base when you were doing the acid and base work last week?
All right. How we doing?
That's so cool.
You already have DNA. That is your DNA, that white cloud there. Do you see that? See that? Now, well, you know, it's just such a big molecule, so we're not going to see the double helix that easily, are we? But the longer we wait, the more product of that DNA we will get. The compounds you put together are actually extracting. They're stripping the DNA molecule of its proteins, and they are pulling it out of the cells, and now you're going to be able to spool that up in a little while. All right, cool. That is awesome.
Funny the things you can find at home, huh? You can see your DNA. Pretty crazy.
All right, why don't we have you guys come back on up here, and we'll get the next part ready. If you girls are all set, we're going to go to the next part while we're waiting for our DNA to finish out, okay? So Miss Crystal's going to get you started. Crystal, I can do this part for you if you want to. Okay, we'll grab the kids. You got it? Isn't that neat? All right, guys, we're gonna come up and have us do one more thing here. Oh. Here. Crystal.
Did you know that you can eat yourself?
Yeah, I know. That's kind of gross.
Cheat
I don't know if I like that.
Can I put it upside down?
Every time you have a meal, doesn't it? How do you guys feel about that? Is that good? Yeah.
You can reprocess
Isn't that
it.
cool? That works really, really well.
I feel extra when I said that.
Was that kind of cool?
Now I think
Yeah?
Crystal's going to continue.
All right, we're going to let those sit. Then you guys are going to see something really cool when we come back, all right? The next part that we were talking about is called DNA Alias, all right? You guys are going to be DNA detectives after I give you a little bit of information about DNA. There are 4 letters up there that code all the information contained in DNA. Those letters are A, T, C, and G. All of these letters are used in groups of 3. Doesn't matter which one is grouped in it, they just are in a group of 3. So each group equals a codon. A codon This is O-D-O-N. So one group equals this. All right? Codon. Can you guys say that? Codon. All right. So DNA contains information needed by your body to make proteins. Each protein has a specific function, such as your heart. What else do you think? Lungs? Liver? Stomach? Yes, or your hair, or your eyes, or your ear, something like that. And then the smallest part of those proteins are called amino acids. We're just going to be using that. I just do AAs. All right? Those are amino acids. There are 20 amino acids. One or more amino acid can make up a particular protein depending on the specific protein. So since each amino acid is represented by at least one codon, because each codon contains 3 letters. Are you guys tracking with me? All right. These string of letters that are used to represent amino acids can get really, really long because you got to use all those letters up there. So to avoid this, scientists have come up with a shorthand in order for them to figure out what amino acids the letters are. So each letter in our alphabet each amino acid has a letter in correlation to our alphabet. That's called a spelling, this part of a protein. So written in shorthand, the code is called a DNA alias, which is what we are going to try and determine with you guys. So when scientists see DNA of a particular protein, they can find DNA sequence by reversing, there goes my pen top, by reversing the coding process. So now for fun, we can find our proteins of DNA by using a sequence and reversing the coding process. So we are going to perform the same thing as scientists do by using any word to convert to each letter or to their corresponding codon. So we're going to use that with your guys' name, okay? So like I showed you earlier, this is my name spelled out with the letters, okay? So these are the packets that we're going to give to you. And your job is on the other sheet of paper that you're going to get. So write your name out. And when you find the letter of your name, it's going to have 3 corresponding codons that are next to it. You need to write that group of 3 in the blank that you're receiving right here. So such as, she's going to put her name right here. And then, so she's going to find the letter here. So your name's Gabby, right? All right, so we're going to find G. So she's going to write here G, G, G. Because G, glycine, is G, G, G. Does that make sense? So you're going to go through your whole name, and there's going to be, for each letter of your name, there's going to be 3 letters that you have to write in this blank. Okay, so there are going to be a lot of letters depending on how long your name is. Yes?
Should I use my whole name, which is Maximilian, or just Max?
Max would probably work. You can choose whatever you want. If your names are shortened, then you can use their shortened name or your full name. It's just going to depend on how many beads you have in your bracelet. Put it that way. So if you want a bigger bracelet with a lot of beads, then put your full name. If you guys have questions, then.
So do you have a pencil, honey? Okay. All right.
Yep, you guys got it. Are you going to do your full name or your? Okay. You want to write your full name here so that you can, it'll help you remember what letter you're on. I had kind of fun with this too when I was doing it at home.
Histidines is T.
Cat.
C and a T is cat. Look
You have a cat in your name.
Look at H.
Oh, yeah.
I'll get one of those.
There's H. Oh, it says cat. Then there's gag. There's gag.
Yeah, there's some goofy ones in there, aren't there?
There's gag.
Gag me.
There's also E, that's gag. Wait, where's E? Oh, gag. Then
There's tack, like tic-tac-toe.
There's gat.
And then ACT that you're going to have to take when you get older.
There's cat for cat.
All right, when are you done? Okay. So when you're done, look up at the board and those letters right there. So you have a G. A G is going to be blue, so that means you're going to need a blue bead. Okay, so you're going to go through with each of the letters and look up at the board and use that color bead to make your bracelet. Okay, and then that's your DNA.
But what about like I have other letters than A, T? What?
Oh, that's fine. Okay, let's look at here. So we have G, right? So G up there on the bottom is going to be a blue bead. And then we have an A, which is going to be a green bead. And then we have C, which is going to be a red bead. So these represent those colors up there. You understand that now? Awesome. Okay.
I take 2 whole rows.
You did take 2 whole because you used your full name, didn't you? All right.
A row and like.
All right, does everyone understand now what you guys are going to do with the beads? Okay, and there's these beads, or I have these beads, okay? So you can choose whatever one you want. These ones right here, I know they don't look red, but you need to use your imagination. They're kind of like peachy. So it's just kind of a cool bracelet that you guys can use or if you want to wear sometime else. And these other ones are kind of like shells. They have black on the back of them, so that's our black, all right? So if you use these, the pearlier white ones are your red, and the shells with the black on it are your black, okay? So if you guys are all done and you're done writing that, let's go to the tables and let's work on it. We got some black wire.
Genetic code ready to go.
Do you want us to spool out the DNA first and do that later?
I think we'll wait.
Okay.
Yeah.
Oh my god.
Oh, wow.
That's mine.
It's coming out.
That one's mine.
That's pretty cool. Don't touch it. We got to keep it awesome. All right, there you go.
I've got a ton.
And there is yours, sir. You get to make a bracelet. So you guys get to use what color?
Crystal,
So
do you have one more spool or
one more what?
one more string?
Oh yeah. I think over there is my.
Oh, here we go.
I'm not sure if there's any.
Well, that's okay. You can get started. I'll find it.
I think Laurie had it. Did you have the right there? Yep, here she has another one.
Okay, so
Okay.
do you like.
So you have a T. So look up on the board and what color is the T? If you can see.
T is green.
T is actually black. See, T up there. A, T, C. The T is black, and then your G is blue. So you're gonna have a black, a blue, and then is that a C?
Yes.
Okay, then you're gonna have a red. So you're gonna do it just like that with each color. And what helps with these guys is if you just hold the other side. Or he got a knot, there you go.
Wait, what? How do
Here, I'm gonna make this a little bit easier to read. So this is my bracelet of that. So that's what my name is gonna look like. And those beads. All right.
I need GC red.
You got it over there?
Chris, you got the red.
I don't know if that's gonna stay. Maybe if you hold both sides of it, hold that side and then put your bead on this one. So just hold this in this hand and then start putting the beads on there. Yeah, with both your hands. Unless you want me to hold it and help you, but. G, G, G. Oh, you got it easy. You got a lot of blacks? Depending on your DNA code, they're all different, huh? See, I don't have hardly any blacks on mine. Well, I do have a few,
Look,
but
I've got 2 blacks in the first.
I've got to do mine. This is really cool. Did you figure this out? Did you find it?
well, we had done it before, and so, yeah.
It's cool.
Yeah.
Cool. It's a cool fancy.
Yeah. And if you guys need any help holding them or something, or
I've got.
it'll let you know what you got. You got a lot. Do you want some help? It might take you a little bit.
I've got a black, and I think.
What one do you
everyone
want?
has got, I'm working on that one.
You're working on this one. Do you have the T?
Yeah, so I just put that on.
So you need the A, which is green. 2 greens. Get 2 greens.
And then I need a black.
And then you need a black. 2 greens, a black, and then another green. And then another green. So,
Seriously?
yes. So after that black, you'll need to put 2 more greens
that's
on.
looking great. So it's uniquely you. Even though all 3 of you share 99.5% of the same DNA, you all have your own look. Look at the way you look different. Look at the way your bodies look different. Even though all your DNA is so much the same.
And then you got a black and then a blue. And you won't be able to fight over whose is whose because you guys will each have your own bracelet.
So just like our DNA has just 4 bases, you're looking at just
And then green, black, green. You're kind of getting the pattern, aren't you?
Please look at this, green and black.
Do you like green and black? You do now, huh?
I prefer red, but.
All right, and then after that, you get a red. You get a red, black, red. And then green, black, red, black, red.
Really? I got a lot of beads too. Look. They're just not all beads.
And then you got a green, black, green. You're almost done, dude. And then a blue, red,
Okay.
blue, red, black. Blue, red, black. And if you want to finish the last one, you're golden. We flew through that one, huh?
Red.
looking good.
Yeah.
Wow, look at that sequence.
It's got a lot of green right there.
That is a wild sequence.
I hope you like green too.
Oh, down it goes. Dang it. Well, I think you can salvage it.
We'll get
We
it.
just got to be sure we don't pull it up.
Man
pull it
down.
from the edges.
At least you have a knot on the end of it.
Yeah, that was smart.
All right, let's go back to where we
Okay, so and
The
the last
pattern.
thing is a black.
find that blue one. The blue is G.
The last
So
thing I've got is black.
G. Did you think it went all the way back to there? Were you almost done?
Blue is G.
We have blue,
Green, black.
blue, green. Does it go blue, green?
It goes blue, green, black.
Blue, green, black. Okay, so
It must be
blue,
right there. So now I
right
need
there.
to
So
add-
now you need the red. So hold it over the
No, wait,
table.
but then no, it goes blue, green, black. So I need to add the
Red. Oh, the green. Okay, yep, you're right. I want to hold over the table or over your thing. How you guys doing? You want help here? Let me tie it. Do you want me to tie it? Do you want it on you, or do you just want a bracelet or something like that? You want me to put it on you?
Sure.
Okay. Because I don't.
What happened to that?
Let's see if it'll fit. Maybe it'll fit on your wrist right like that, because there's kind of a knot in it.
There.
Do you want it tighter? I can make it tighter. Let's hold it over this plate so I don't drop it. I don't want to do that.
Alright, it goes.
What happened? You took it all off?
Because I messed something up.
Uh-oh. I'll come help you in a second. All right, is that too tight?
No, it's good.
That's good?
On the way. She is.
Okay, and then we can cut off the end of that. I got a scissors, so it won't poke you. Okay. I'll go get one. How's it come over here?
Okay.
We're doing really
Oh, these are looking
well.
awesome.
I think they look great.
Wow.
All right, come apart.
Cool, and if you guys need more colors of something, I have more.
Yours is like all green and mine
Oh,
is
those
like
look
all
awesome. And if you guys need more colors, I have more, so don't be afraid to ask. All right, let's cut you off.
Okay, so you want the bottom cut. Can you do that?
You're welcome.
This is so cool.
I know, haven't looked at those lately.
I love it. Some people are using theirs as a backpack key holder instead. Or you could use it as a bracelet, either one.
Nope.
You could do it as a bracelet, or you could keep it straight as a backpack holder. Look at yours, how cool it is.
All right, do you need any help? Which one you want?
T, which is black.
All right, how are we doing, everybody?
You're on this one?
Done?
No, here.
You're right there? Okay. You got 3, so you're on that one. So you need 3 more. All right. Oh, you got it tied on too. You got it tied on good? Okay. So I don't want it to open up. And you want me to cut off the ends in just a second?
No.
You're good? Cool. Those look great, guys. And then green, black, green.
This isn't actually the code in your body, though, is it?
Do your name. Code for your name. Green, black, green, and then blue. Blue, green, red, right?
Yeah.
Or is that it? Yeah. Okay. You want me to put it on your wrist?
Sure.
Or do you want it just kind of tied?
What happened to all your greens? I accidentally added
Oh,
green
that's right, you
instead
did.
of black.
Did we get them all on? Current's got his.
That reflects your
Max
personality.
got his.
Black, evil, right?
You like the punk type
Oh,
stuff.
that's sharp. Oh, very nice.
Well, I got a lot of
All right, let me cut those off.
Look at that massive band of black right there. Yeah. That's cool.
Let's take a look at everybody's. Let's kind of gather back to the circle up here.
You want to take paper?
This time let's line up half on one side, half on the other.
That would be awesome.
Okay?
And my paper's over here. Maybe they're not. Yeah, they're up here.
All right, we're going to line up like a quiz bowl.
Quiz bowl.
Let me see. You want it tied on your wrist? Or are you going to use a keychain?
I know my wrist, but I can't tie it on myself.
You want me to tie it? Actually, I think Mrs. Fott, you want to tie some of their bracelets? We'll get going with this game. All right, I'm going to need, hey, Gabby, would you like to be on the gentleman's team, or would you want me to choose someone else to go over there? We want to be over there? Okay, that would be fantastic. All right. All right, you guys ready? We're going to play a quick game because you guys are going to have to leave soon, okay? So let's come up close to the table. Can you girls come up close to the table? So you got to be able to hit the table, okay? All right. We're first going to start, it's going to be between these 2 girls first, all right? I'm going to ask you a question, and if whoever hits the table first, all right, they're going to have a chance to answer the question. This is going to be like Family Feud, okay? But this is going to be DNA Classroom Feud. So, you want to keep score? That would be fantastic. Okay, so you girls ready?
What's your name? What's the team's
Okay,
name?
keep your hand off the table, okay?
Yeah, what is your team
Yeah,
name?
we need team names.
We are DN and you're A.
What?
We'll let.
We're the N.
You want to be DN? No.
I don't.
Or DNA?
We're A pluses. I was just kidding.
A plus?
They can be
We're the A
RNA.
plus.
No, you guys are RNA. We're
Okay, the quicker we go, the quicker we can get more questions in.
you are DN and we're
Okay. All right, DNA versus the A pluses. Here we go. Question number 1. Whoever hits the table first gets to answer it. How many cells is your body made up of?
200.
It was in the movie. Do you have a guess? Okay.
Something trillion.
Something trillion. We'll throw that one out because
4 trillion. 5 trillion.
close. You need to add a
6 trillion? 3 trillion.
Nope.
It was 50 trillion.
50 trillion. Okay. Nobody's going to get that one because we didn't get that one. So 50 trillion cells. Okay. All right. You two ready? Inside every cell is a nucleus. 1 point for the A pluses. All right. Question number 3. Are you ready? Okay. Every nucleus contains what percent? You're going to finish the question for me?
No, I don't know.
Okay, then don't hit your hand until I read it. All right. Every nucleus contains what percent of your genes? Ooh, do you know?
I think it was.
I think
It sounded like
rock, paper, scissors. Go.
Rock, paper, scissors.
Rock, paper, scissors.
rock, paper, scissors.
All right, she got it. Okay, what is your guess?
Wasn't it 98%?
I know what it is.
So close.
99.9%.
99.9%. We got it. You were so close. All right, next. You guys ready? How many genes do we have?
I know this.
Then tell me.
Think, Karen, think.
10 seconds or else it's going to the other team. 10, 9. Nope. You have a guess? Right, whoever slaps the table first.
A million?
Nope. I think we're going to throw that one out too. Yep. 20,000 genes your body has. Okay, next. You girls ready? Okay, hands off the table. Your genes are part of what?
DNA.
DNA! Woohoo! Somebody who's listening. Yay! Okay, next. What does DNA stand for?
Deoxy.
Try it.
10 seconds.
Deoxyribo.
All right, we're going to go over here. You have a guess?
Deoxy was the nuclear acid.
Yeah! Woohoo! Deoxyribonucleic acid. Yes, she did.
2 to 2.
All right. This has 2 different parts of this question. Okay? DNA is composed of blank and blank.
Like genes or 2. I know. Okay, I don't know.
2 strands.
Nope.
Don't look.
Sugar and phosphate. All right?
I knew it.
We're going to give you guys another chance, okay? How many different bases of DNA are there?
4, right?
What are they? Don't look at the board. Don't look at the board.
A, T, C, and G.
All right, there we go. Okay, next one. This is for double points, okay, you two?
Whoa.
Name one of the words that represent A, T, C, or G. Like what the word, what the letter, what it actually.
Oh no. Let me guess.
That's why it's double.
So you're.
The name for them.
A special name for them.
There's like
5 seconds.
I don't know.
All right, do you have a guess? Does she get one?
What is it again?
Say it again. Oh, close. That's pretty close. Should we give her 1 point? Adenine. Adenine. All right.
3.
All right, here we go. Those 4 bases up on the board spell out our blank code.
Name? I know what it is. It's our genetic code.
Genetic code. Ah, nobody's got that one. All right, that was so close though, but I needed genetic. All right, you two ready? DNA is organized into pieces called.
Did you know this?
Or just want to hit your hand on the table?
I think you know, you just want to get the first guess. It's
5 seconds. 5, 4, 3, 2, 1. Oh. Chromosomes.
So it's still 3 to 3.
All right, here we go. Humans usually have how many chromosomes?
I have no idea.
Deduction of a point. I'm just kidding.
Wait, is it-
Do you have a guess?
I know what it is.
This was all in the movie. Come on. No, I'm just kidding. It went really fast.
It was. He knows what it is. I think it was 46.
Yeah, but we can't give you a point for it. All right, next. We each have 2 pairs of how many chromosomes?
I know.
Shy.
I know what it is.
Don't cheat.
Stop. Griffin.
5 seconds. 5.
24.
No.
47?
No. 23. You were so close. All right. All right. Back up at the front. A, T, C, and G are used in groups of how many?
3.
3. We got another point. All right. Each group of 3 is called a.
Don't look.
Use your resources. Do you have a guess?
Wait, what was the question again?
You guys should get this one. Each group of 3 is called this.
Griffin. I know it.
A codon.
Yeah, codon.
Yay!
Acids.
Okay, throw that one out. Next group.
4 to 3.
Last question. Okay, let's find a good one here.
We gotta get this. It will be tied.
All right, DNA. Here we go. DNA contains information needed by your body to make this.
Proteins.
Protein.
That's tiebreaker.
Oh.
Gosh.
All right, here we go.
Well, that was just
How are we doing on time?
It's 44.
One more. Okay.
can do this. Come on, current.
Each protein has a specific function. I didn't even read the question. Name one of those possible functions that was mentioned in my lecture.
I think I know this. I might know this.
Each protein has a specific function. Name one of those possible functions that was mentioned in my lecture.
Like
Lecture.
like what you can get.
Couldn't think of another
DNA
word.
from? What proteins do?
Okay, we're gonna throw that on. Heart, eyes, ears, or hair?
Hair.
Yeah, good.
Yeah.
Okay, this
for that, Curran.
we are going to. This is the tiebreaker. Okay, right here.
Oh, no, I don't.
Here we go. What is my first and last name?
Oh,
Crystal.
that's my hamburger. Crystal hamburger.
Very good.
Crystal. Yeah. Do you know it?
I'm not sure.
Should have listened, huh?
It's so hard to remember.
I know. That's why it's the last one.
Crystal something.
Oh, very good.
Crystal, does it start with an H?
I thought it was.
Okay, we're gonna go to the next people in the group.
Oh dear.
Does anyone know? Do your hands if you know.
Crystal.
Does anyone know?
He just went like that.
It doesn't have an M.
That's the closest we've had. Should we just leave it as that?
We should.
Okay, Crystal Meekoff.
Oh, yes. See, I knew it was
There we go. All right. All right. Here we go. You guys each get some candy to get hyper for your teachers, okay? All right, and then the winning table, you guys get one of those. Well, you guys get each one, and then they give double.